Hello guys, welcome to the master class. We are continuing our exercise on the important essay type questions on applied microbiology for university exams. As you already know, I'm explaining the essay type questions, and within an essay type question there are different other questions also, like short, very short, and multiple choice questions that I am explaining in these classes. So, if you are watching this video for the first time, you must watch the previous videos for different questions. So, let's come back to today's session. So, guys, the next essay type question is define culture media, describe different types of culture media with emphasis on special media. So the question has asked to give a definition, but where the definition is not asked in the question, I have said you earlier, you have to give an introduction at the beginning of the essay type question. But here we have to give the definition of culture media. So the definition is culture media are nutrient preparations used for the growth, isolation, means separation, identification, and maintenance of microorganisms in the laboratory, because many times the microorganisms are preserved in a laboratory. That's why we have mentioned maintenance of microorganisms in the laboratory. So this will be the definition. So let's start. Give the broad heading: classification of culture media based on composition and function. So start with solid culture media. This media contain a solidifying agent, usually agar. Agar is basically a complex polysaccharide, 1.5 to 2%. Characteristics: firm, means solid, firm surface for microbial growth, used to isolate pure colonies, help study colony morphology, because colony, each and every colony has a particular morphology. It is studied here. Pigmentation, because the colonies and the microbes, they produce different colors, that is called pigmentation. Hemolysis, when put to a particular culture, especially the blood agar, the bacteria produce the hemolysis. So these are the characteristics. Examples: nutrient agar, blood agar, Macanki agar, and chocolate agar. So guys, solid culture media, we are continuing. Mark this particular question, solid culture media, is a 3 marks question. Mark it. So uses of solid culture media: isolation of bacteria, counting colonies, antibiotic sensitivity testing, very important for MCQ, underline this. Identification of microorganisms. Advantages: easy to separate mixed organisms, because in many cases different types of pathogens or bacteria particularly, they are found. It is easy to separate them. Colonies can be observed individually. Disadvantages: preparation takes more time and not ideal for large scale growth. Next, liquid culture media. Again, a question for 3 marks. Make a note of it. So definition: liquid media do not contain agar, and are called broths. Underline this MCQ. Characteristics: nutrient dissolved in water, microorganisms grow throughout the medium. Example: nutrient broth, most commonly used liquid media. Underline this for MCQ. And the composition of nutrient broth is peptone, beef extract, yeast extract, sodium chloride, and distilled water. After nutrient broth, peptone water, glucose broth. Uses: rapid multiplication of bacteria for biochemical tests and preparation of inoculation. Underline this for MCQ. Inoculation means intentionally the microorganism is introduced to a culture medium to observe its growth. So the advantages of liquid media are supports abundant growth, because when a large amount or quantities of bacteria need to be cultured, during that time liquid media are used. Easy to prepare. Disadvantages: difficult to isolate pure colonies. This is possible in case of solid media. And contamination, because most of the time or sometimes the contamination that happens is difficult to detect, because throughout the medium the turbidity will be there, so sometimes it is difficult to detect the contamination. So this is all about the liquid culture media. 3 marks question. Like solid culture media, also 3 marks question. Next, semi-synthetic or complex media. Mark it. 2 marks question. This semi-synthetic media contain natural substances whose chemical composition is not known. They provide nutrients required for the growth of many microorganisms. Examples: nutrient broth, we studied in the previous slide, nutrient agar and blood agar. Uses of semi-synthetic or complex media: routine cultivation of bacteria. On the day-to-day, in the normal practice, that is called the routine cultivation of bacteria. In those cases, semi-synthetic or this complex media are used. Next, synthetic or chemically defined media. 2 marks question. Contain pure chemical substances in known quantities. Underline for MCQ. The exact composition is known. Example: look at the spelling. Zapic tox medium. Underline this for MCQ. Minimal salts medium. Uses of synthetic media. Research studies on bacterial nutrition and metabolism. Guys, if you are watching this video on a mobile phone, please watch in a full screen, because full screen watch will prevent your distraction. So first you watch or listen the whole explanation, then mute your audio, then start writing the points, and at the end you will find your notes is ready, so you can do in all the videos available in this channel. So next, special media. In that essay questions, already I told you that on special emphasis on special media. Here, look carefully. This only special media is a 5 marks question, because under special media so many media will be there. Now to start with, within that special media, enriched media. Look, only this enriched media is a 2 marks question. So you note this mark distribution and prepare your notes accordingly. So enriched media, these are base media supplemented, means added. One base medium will be there, some other things will be added, like with blood, serum, egg, other growth factors to support fastidious organisms. Fastidious means difficult to grow, because certain microorganisms, they have some very specific nutritional requirement. They are known as fastidious organisms. So they are, this enriched media is used for that. Example of enriched media: blood agar, chocolate agar, Loeffler's serum slow. Check out the spelling here. Uses of enriched media: cultivation of fastidious bacteria. Underline this for MCQ. Such as Streptococcus species, for example, Streptococcus pneumoniae; Neisseria species, for example, Neisseria gonorrhoeae. Guys, you must have observed how to find out the short, very short, and multiple choice questions within an essay type question. So you make the note accordingly and prepare your notes accordingly. So next, in special media, enrichment media. Earlier we covered enriched media. Next is enrichment media. Note it. This particular enrichment media is a question of 2 marks. So they are liquid media that enhance, means increase, the growth of a desired pathogen while suppressing unwanted organisms. Understand the concept here, because when the culture is made, what happens? There is a possibility of mixed pathogens. Suppose we want to grow particular bacteria A, but along with that, bacteria B, C are also present. So what will happen? This particular media is a very targeted media. They will ensure that's a desired means what exactly we need or require. This media will grow that particular organism and it will suppress the growth of other bacteria. And this media, enrichment media, is used when the pathogen is present in small numbers. Examples: selenite F broth, used for Salmonella species like Salmonella typhi, Salmonella paratyphi. And the second example in this enrichment media is tetrathionate broth. Enrichment media is continuing. The next example is alkaline peptone water, used to isolate the bacteria Vibrio cholerae. Use of this enrichment media is isolation of pathogens from mixed bacterial population. Underline this for MCQ. As I told earlier, when there are different types of bacteria mixed, and we need, that is our desired pathogen, we need to grow one particular bacteria, we use enrichment media. So underline this for MCQ. Isolation of pathogens from mixed bacterial population. So guys, next in special media is selective media. Mark it. Selective media is a 2 marks question. Now, what is selective media? This media contain inhibitory substances that suppress unwanted microorganisms and allow the growth of specific organisms. This will contain certain substances that inhibit the unwanted microorganisms. Guys, there is a possibility of confusion in enrichment media and selective media. Let me clarify it. Suppose there is a group of bacterium A, B, C are there, mixed culture, mixed bacterial population. So in case of what happens, in case of enrichment media, suppose in that case our desired bacterium was bacterium A. So the enrichment media becomes favorable or helps to grow bacterium A, so that bacterium B and bacterium C will not grow. But this is something different. In selective media, same bacterium A, bacterium B, bacterium C, 3 different bacterium or bacteria are there. What happens? This media, suppose our desired bacterium is bacterium A. This particular media, selective media, what this will do? They will totally inhibit or stop the growth of bacterium B and bacterium C. So in that case, our desired bacterium A will grow automatically. So this is the difference between enrichment media and selective media. I hope this is clear to you. Now, examples of selective media is McConkey agar, that inhibits gram-positive bacteria. So, suppose there is a mix of gram-positive and gram-negative bacteria. So, in this case, what will happen? Automatically, the gram-negative bacteria will grow automatically because McConkey agar inhibits gram-positive bacteria. Next, mannitol salt agar, selective for Staphylococcus species. That means this will inhibit all other microorganisms so that Staphylococcus or the species like Staphylococcus aureus, they can grow. Similarly, Lowenstein-Janssen medium is selective for Mycobacterium tuberculosis. So when this media is used, only Mycobacterium tuberculosis will grow automatically and all other bacteria present in the mixed population, they will be inhibited, their growth will be stopped. So uses of selective media: isolation of particular bacteria from mixed culture. So next in special media is differential, also known as indicator media. Again, 2 marks question. This media contain indicators, means they contain certain chemicals. What will happen? That help distinguish microorganisms based on their biochemical characteristics. Because some of the bacteria, they have some bacteria biochemical properties, and this indicator in this differential or indicator media either will produce some color, or something else that we will know here. Example, Macanki agar. When this Macanki agar is used, what will happen? The lactose fermenting bacteria, they will produce pink colonies. The colony will look pink. But in the same case, in that mixed culture, if non-lactose fermenting bacteria or fermenters will be there, they will produce colorless colonies. So both lactose fermenters and non-lactose fermenters can be separated from this color difference. Pink colonies, colorless colonies. But this is one part, color. Next comes something else. When the blood agar is used, that shows hemolysis pattern, because in certainly RBC are there in blood agar, so there will be hemolysis pattern. Bacteria can be identified from that. Hemolysis pattern, alpha, beta, gamma, hemolysis pattern. So this differential or indicator media is used for the identification and differentiation of bacterial species. So this is all about our special media and the whole explanation of the essay type question. So guys, if you like the videos, give a thumbs up here, and please drop your comments and feedback in the comment section. So guys, keep watching and keep learning and keep growing.